Transposable genetic elements, or sometimes just called transposons, are small pieces of DNA that can insert themselves and excise themselves from chromosomes. Sometimes they're called the jumping genes. Barbara McClintock won the Nobel Prize in 1983 for her work in the 40s of transposable elements. Transposable elements are common in bacteria, where they're called insertion sequences, and then also in maize, that's where Barbara McClintock did her studies, and Drosophila, for example. So there's a YouTube video here that I will link up. If I play it right here, you won't really be able to view it very well, but I would recommend that you pause this lecture and scroll down on the page and go ahead and watch this short YouTube video about transposable elements in plant evolution. So the way a transposable element works is it typically on the transposable element itself, on the DNA of that short segment, there's a gene for an enzyme called transposase. And that enzyme will cut the host chromosome typically in a staggered pattern, and then the transposable element inserts itself into the chromosome, and then DNA polymerase fills in these little gaps that are created, and you end up with what are called direct repeats, and that typically signals where a transposable element is located. It's one of the things that you can look for when you are looking for transposable elements in DNA. And this is just a close-up of a transposable element. Typically, the transposable element itself has repeated sequences at the ends, and then the place on the chromosome where it inserts, because of the DNA repair that has to happen, will have direct repeats. So on the bottom image, they just show you a direct repeat is the same repeat shown in purple. It goes the same TGCAA, for example, here, and then TGCAA, for example, here. Whereas an inverted repeat goes ATCGCA, for example, on this strand, so going from left to right on the top strand, you would have to go from right to left on the bottom strand, ATCGCA, to see that same sequence. So that's called an inverted repeat. Now, one kind of easy example of how transposons create different alleles or different phenotypes is that in grapes. Black grapes have a functional gene that's that creates the production of anthocyanin pigments, which makes the grapes black, because that's really the natural color. But a transposable element or transposon inserted near to the gene and interrupted the expression of that gene, and you get green grapes. But then if the transposable element excises or cuts out of the chromosome and leaves, then you end up with a piece of the transposable element that's sort of interrupting the expression of this gene, so you get some expression of the pigment but not the full black color, you get the red. So it's just an example of how transposons jumping into a chromosome and then jumping out of a chromosome can create different phenotypes. Transposons can jump into genes directly. That creates mutations. They can jump near to genes, like this example, where it just creates a change in gene expression. So an interesting question that we've always wondered is, are there human transposons? Well, we do have this sequence called ALU. ALU is a repetitive sequence. If you remember in the chapter when we discussed repetitive sequences, ALU is one of the group of moderately repetitive sequences called SINE. S-I-N-E stands for short interspersed element. And ALU is a 200 to 300 base pair sequence that appears more than 1 million times in the human genome. And what we think it is represents sort of an ancient transposon, sort of a viral jumping gene that spread through the genome, copied itself many times, and then inserted itself in the genome, and then at some point in evolution it lost the enzyme that it needed to be able to cut itself out of the chromosome and move somewhere else. So now the ALU sequences are stuck on the chromosomes. There is another repetitive element called a line, long interspersed element, that was found inserted into the clotting factor gene of 2 hemophiliac patients. So it was the insertion of this very long interspersed element created a gene mutation because the gene was completely interrupted. So these individuals cannot make a certain type of clotting factor, and therefore they have hemophilia. But what's interesting is that none of the parents of these individuals had the gene that had that line sequence inserted into it. So what that means is that the transposon must have actively moved, during gamete formation or in the early embryo, for example. So there must have been an active transposon in these individuals very early in development or in gamete formation. And so that kind of changes our thought, because looking at ALU, we think of human transposons as being sort of relics or remnants of things that can no longer move, but maybe millions of years ago were able to move around in the genome, but now they're just stuck. But then you see this evidence that there's an active sequence, an active line sequence inserted into the gene that causes hemophilia. So that kind of changes our view on it, in that we have to admit that there have to be at least some transposable elements that are actively jumping around. So that could create new genes and new alleles. We don't really know how active these elements are in human. And I put one of the links that you could, if you're interested in this case, you could look it up.